Good morning, everyone. Thank you, Chantal. Thank you very much, Lama, for this invitation. It's a big honor to be here. Thank you, President and Deans, and all the speakers. I also want to acknowledge a special guest in the audience for me. It's my mother-in-law, Mrs. Aida Hanoun. She's a brilliant mathematician herself, and luckily I have 3 brilliant mathematician kids because of her. So one of them actually is studying artificial intelligence and hopefully will be contributing to the field. So I just start the slides with, okay, so today I'm going to spend time just very briefly to introduce to you some concepts that many of you probably know a lot more about than I do, but, oh, it's going up, okay. Maybe the lights, can we dim the lights just a bit so you can see? Okay, perfect. So I titled my talk Data-Driven Science, and I primarily said applications to life sciences. And so you all have already heard, and you'll hear a lot more today, that we are in an era of a digital revolution, and the results are big data. Typically, the word big data refers to data sets that are so large and so complex that they are difficult to process using traditional applications. And it has the potential to impact almost everything, as you've heard already, and you'll hear a lot more today, including travel, shopping, banking, education, sports, but something very near and dear to my heart, life sciences, is no exception. So the 3 things that also I'm sure many people who work with big data know are the 3 V's. Not only do we have mountains of data, which refers to the volume of data, but also we have the velocity of processing the data, and that's a crucial thing. So you have these mountains of data, but how fast can you learn these data, process these data, and use these data? And also a very crucial thing, particularly in the life sciences, and difficult to wrap your head around, is the variability of data sources. And I'll give you some examples that apply to the life sciences. But as you know, the variability of data sources, including social media and all kinds of other sets of data, are really immense. So in the life sciences, I just have a brief set of examples because there are incredible amounts of examples of different data. But for us, our main goal, maybe I should start by the bottom point on the slides, that our common goal is to generate better outcomes and improve health for people. And so in order to do that, we have access to lots of data. Population-based data. So we're able to gain data from populations. And actually, one of the important goals of the National Institute of Health now is one of these programs that are called Centers for Translational Science, which I'm in charge of building one at Stony Brook in collaboration with several other senior faculty and chairs and deans, is to build the Center for Translational Sciences. And the goal of these centers is indeed to make every patient ever seen be a piece of data. I mean, not in a negative way, but benefit from data from every patient you see. So potentially every patient that is admitted or seen as an outpatient or records anything in a drug in a pharmacy has to be considered data, and that person, or you can use these data then to mine them, to understand outcomes, to understand what can be helped and what the population health needs, etc. So these population-based studies start with things like electronic medical records, EMR, so keeping very thorough and good electronic medical records, and is an important revolution and something that's actually not as well done as we would like it to be, because electronic medical records are not just the ability to keep records, but the ability to mine these data and be able to make sense from all the noise that you get. And as you can imagine, every piece of information that your physician takes from you, including every blood, every time they touch you, every time they feel your blood pressure, your pulse, all these are incredible pieces of data. And every time they take an EKG for you, that EKG has tons of data. Every X-ray they do has lots of pixels. Every piece of pathology can be examined under microscopes and taken as multiple pixels of data. So you can imagine the mountains and mountains of data that we can gather and that we are still not completely taking advantage of, but we will have a lot of knowledge. Obviously, healthcare plans, web applications, because people now use web apps for everything. They check their blood pressure, they do other life-related stuff on their web apps. Then there's the pharmaceutical industry, which generates a lot of data, research and development, areas that I'm interested in. And that's all at the macro stage. Then we go to the more molecular stage. Molecular pathways are incredible sources of generating data. And digitized information from genomics, the revolution that you hear about in genomics, where everybody has their DNA is sequenced. Now we can have the whole human genome sequenced for $1,000 in less than a few hours, which generates incredible amounts of data that can also be utilized to personalize medicine. Then there are all these new terms that you might or might not have heard of. Pathomics, our ability to look at pathology and mine data from it, like I said, looking at slides or any pathology from patients. Radiomics, the ability to mine data from all radiology, x-rays that you take, and other different, and you'll hear from actually one of the speakers today, Dr. Anissa Dargam, about PET scanning and very interesting approaches, again, generating a lot of new data. Then proteomics, which is the ability to understand and dissect proteins at multiple levels. And I'll give you some example of those because they're some of the things that we do in my lab. Metabolomics, the ability to understand all metabolites, and there are thousands of known and thousands more of unknown metabolites that we're able to access by what's called shotgun mechanisms of mass spectroscopy, mass spectrometry, where we're able to find all these pieces of data and then try to make sense of them. Again, lipidomics is an area that I work on as well, and that's the ability to mine data from all lipids and understand lipids and their roles in health and disease. So a bit about my university. Lama asked me to speak in the capacity of Dean for Research, and Stony Brook University is a really exciting and interesting place because it is a main university similar to AUB, and it has a medical school with it. And I think what you have at AUB is also a really beautiful combination of the two, because you're able to cross-fertilize each other. So at Stony Brook, the campuses are very close to each other, and it brings together amazing strengths in computer science, applied mathematics, statistics, bioengineering, chemistry, physics, computational biology, and the life sciences, including basic and clinical areas of study. And this has really allowed the cross-fertilization and interaction between all the above, making Stony Brook a very, very poised to tackle the explosion of big data and life sciences. So it has been very exciting for me to watch this happen and to also partake in growing it. And I'll give you a few examples. So when I arrived to Stony Brook, there was a major strategic plan underway, and I highlighted some of the main areas of the strategic plan. They were to enhance main themes in cancer, cardiovascular disease, neurosciences, and infectious disease. And these are the main, obviously you want to enhance everything, but there were areas that were going to be of specific focus, and they were chosen for several reasons. Cancer particularly because now it is the single most disease that we are still not completely able to treat. And so we really want to attack cancer and be able to cure and treat cancer. But as we discover every day, cancer is not one disease, it's hundreds of diseases, and each cancer is different from the other. And when you say breast cancer, there are several breast cancers. There's hormone sensitive, hormone insensitive, triple negative. So there are, and then for each patient, it's different. They have a tumor that originates in one part versus the other versus both parts. So it's really every cancer, and I can't emphasize this enough, lung cancer is multiple cancers. It's squamous cell, it's adenocarcinoma, there are some genetic markers. So it's imperative on us to understand all these new diseases that we lump under cancer. And to do that, we really need to have big data, knowledge in big data. Cardiovascular diseases similarly, neurosciences, again, the brain and all the neural networks that we have, the brain is actually the gold standard for big data because its ability to understand neural networks is now what people use to understand big data and artificial intelligence. Again, infectious diseases and immunology is also exploding. And so these were the main themes that we chose. And then some sub-themes that also add to what are also critical for understanding disease, and they all use, again, applications of big data, including drug discovery, metabolic diseases. But critical technologies were also needed, and one of them is imaging, because traditionally the university, Stony Brook University, is the place where the MRI was invented, and Brookhaven National Lab, which is a sister lab to Stony Brook, is where PET scanning was invented. So there's a lot of strength in imaging, and because imaging brings, again, as I said, so much data that it's very important for us to understand that and to have robust information technology, because then this will, and I put robust information technology in red because it's going to be important to affect all of these areas that I'm talking about. And then robust research core facilities, which are facilities that are used outside of people's separate labs. They're core facilities that enhance everybody's research. And so if we have them, we're able to share each other's expertise and help each other out. And so that was part of also my mission, is to enhance the research core facilities. Obviously, this was a big and very collaborative undertaking, and we've had several successes. There's been multiple faculty that have been recruited in all major areas of strategic planning, with several faculty focusing on population-based studies, a lot on cancer, a lot in neuroscience. We've also enhanced the research core facilities, including infrastructure for basic and for applied research, and also installed systems, quantitative systems like iLab, which help us keep track of the data that we acquire. And we've significantly enhanced biomedical imaging from molecular to patient-based. But one big accomplishment I was in charge of, I chaired the search committee to recruit and open a brand new department of biomedical informatics. And we were lucky to recruit a chair for that department, Joel Saltz, and actually build a department. And now it's a degree-granting PhD program, PhD-granting Department of Biomedical Informatics, and in the less than 4 years, we've recruited 10 faculty. And this department is very nice because it spans across campus from the engineering side, the computer science side, to the biomedical side, and there's a lot of interaction. And so the faculty in this department really speak both languages, the health science language as well as the language of computer science and data analysis. And so it's really brought these areas together and allows us to grow. So I'm going to give you a couple of examples, and I'm not sure how I'm doing on time. I'll get a warning when I'm close to giving time, but I'm just going to give you a few examples of data. One is that generate huge amounts of data, and that's considered metadata, which is the definition of metadata is data about data, but I'll tell you a bit more about that. But the National Institute, the National Cancer Institute, is a branch of the National Institute of Health. And several years ago, the National Cancer Institute, in collaboration with the National Human Genome Research Institute, started a collaboration which has generated a comprehensive, multi-dimensional maps of the key genomic changes in 33 types of cancer. It's called the Cancer Genome Atlas. And basically, TCGA for short, so far has 2.5 petabytes, if not more so far, of data describing tumor tissue, matched normal tissue, from more than 11,000 patients, and the number is growing. It's publicly available and has been used widely by the research community The data have contributed to more than 1,000 studies of cancer by independent researchers and to the TCGA Research Network publications. So basically this atlas, Cancer Genome Atlas, and I'll show you a bit more about what it's able to do, but it generates incredible amounts of data and it's freely accessible for anyone to take advantage of because it's all de-identified from patients. So one example, this is an interactive example, so it's very difficult to do, but I want to just give you a rough idea of what you can do with this. This is the pointer. Which one's the pointer? This one? Yeah, okay. So I want to say that what's amazing about this is, what basically the cancer genome is that every patient that has come through has had their genomic analysis done, but also looking at their tumor and looking at all available resources from that patient. So so on this side, you can see each circle here. It's kind of a snapshot of what you would get when you do the data analysis. But each circle here, and I know it's not clear, but all I want to point out is how massive it is. But each circle here points to a single patient. And on the on the left side is the age per age and per outcome of the patient. And then there's a set, based on each patient, you know their age, their outcome, and then you know the pathology of their disease according to how much different things have occur in them. For example, is the tumor infiltrated by neutrophils? Is the tumor
This is not working.
Okay. Is the tumor infiltrated by neutrophils? Is the tumor have a lot of lymphocytes? The characteristics of the tumor. And I don't want to spend too much time. All I want to say is that you have massive amounts of data on each patient. And then at the bottom, if you're able to when you use it interactively online, you click on each patient and you know everything about that patient based on that database that's available. But not only that, you can click at the actual pathology of the patient and then you can choose specific nuclei and then study them digitally by taking images of them and studying their characteristics and seeing for each patient, each tumor, what's inside that tumor and what are its characteristics. Does the tumor flow in a certain field? Are there cells that float on their own? And then you can mine all this into massive amounts of data, but at the same time you have the outcome of what happens to the patient. So it's really mind-boggling, but it allows us to, I'm going to turn this off. So it's mind-boggling how much data you can get from each patient, but it allows us to mine these data and gain a lot of information about trends or what's going to be happening to that patient. And that is sort of the era of personalized medicine, or what's called precision medicine, because you're able to understand what happens with every patient and treat every patient as an N of one, while they are N's of thousands, but you're able to take all this data, understand it, and then bring it back to the single patient. So it really is a revolution in care, and I think this atlas is really one of the examples of very exciting. So examples from my lab. My lab has focused primarily on an area of bioactive lipids, as you heard. And my work has been a long-term collaboration with the laboratory of Youssef Hanoun, who also happens to be my husband. And we've interacted very happily and productively, so that we have many approaches. And that's what's nice. We have a group now of 30 investigators, and we have a lot of expertise, and so it's very nice to have this big interactive group because we learn from each other and teach each other, and we have different expertise. So we have used proteomic approaches, which is studying proteins and when they're found in cells. We've also studied metabolomics, which is the metabolites that these proteins make, and we've also studied modeling using also applied mathematics to look at structure of specific proteins and be able to use them to develop drugs. And I'll give you brief examples and then I'll just stop. So one enzyme we work on is called ceramide synthase, and it's an enzyme that makes ceramide. And why are we interested in ceramide? Because ceramide is a very potent inducer of cell death. So we want to understand how this molecule is made and metabolized and its role in cell death. So there are these 6 enzymes that make it, 6 different proteins that make ceramide. Each one has a different chain length or ability to make a ceramide of a different design. So we wanted to understand the differences between them. So we used this approach, which is using big data, not huge data, but big data. We took each one of our enzymes, and we then we made a lot of the protein, and we added a tag to the DNA of the protein, so that when you make the protein is tagged by a piece of protein that identifies it and allows it to be pulled out specifically by itself using immunoprecipitation techniques. But the bottom line is you're able to identify each protein on its own and then subject it to mass spectroscopy, which means you break it into many pieces, and then you run it on this mass spectroscope, and you get according size to mass ratios and how you fragment the protein, you're able to get a very specific map to that protein. And if the protein interacts with other proteins, you also identify those. And there are big databases available that tell you what are these proteins that came down with this protein. So it helps you understand the function of this protein. So when we did that for the different proteins that we're interested in, we get something like this. So for each protein that we're looking at, ceramide synthase 1, 4, 2, each one of these enzymes comes down with different other proteins that they are interacting with. And this is the intensity of the interaction. So red is high intensity, white is low intensity. So as you see, each one of these has a different set of interacting proteins, but some of them have the same set of interacting proteins. So we were able, by using this technology, to define what the different proteins are that interact with each one of our proteins of interest and divide them according to data that is, again, already available in big data sets to tell us if these proteins come from the endoplasmic reticulum, or are they found in a lipid droplet in a cell, which cellular compartment do these proteins exist in, are they in the nucleus, and then we're able to, once we identify the 2 interacting proteins, we're able to validate that in tissue culture, and then able to make predictions. If this protein interacts with this protein, then does it affect its function? And one of our approaches, we found one protein which interacted with many of our proteins, of our 5 proteins, and it turned out to be critical to making a novel metabolic pathway, adding another fatty acid to ceramide and making it a very big fatty molecule that deposits in lipid droplets. And lipid droplets are like big fatty areas that are found in cells. And so it helps us understand now metabolically that when there is obesity, for example, you can turn on this pathway and it has modes of regulating cell growth and cell death. So it's really interesting what we gained from that knowledge. Another example is, again, based on proteomics, but here we are taking we're taking extracts from cells that are coming from, say, a colon inflammation in this case. And in this case, mice were fed a diet very rich in fat, and we wanted to see what happens to the mice. So we obtain the protein from their colon, and then we fragment these proteins, again, run them on mass spectrometry, and able to find what proteins are increased or decreased in your sample of interest. And in this case, we found all these proteins were increased in tissues obtained from mice that were fed high-fat diet. And then when you do a David or string analysis, which is, again, you taking advantage of a database, you can find which proteins interact with each other, and it allows us to find nodes of proteins that are interacting. And through this, and I'm not showing the details, but through this we were able to find that proteins that make cells very permeable and leaky were significantly increased. So it tells us when you eat high-fat diet, the cells become really leaky in the colon. And now we're trying to figure out how that happens and what the implications are to disease and to treatment. A third example, again from my lab, takes advantage of mass spectra, and most of these are mass spec obtained data. And here what we're doing is we're taking cells and again treating them with a mechanism, whatever we want to treat them with. In this case, we're depriving them of a very important amino acid called serine. And the reason we're interested in doing that is because serine is very important for cancer growth. So we said if we take cells and remove serine from them, then they grow less. And we wanted to see how that affects their metabolism. So we take these cells and again subject them to mass spec and look for all the ions that come up in the metabolic pathways. And we found here again multiple different metabolites. And this is a heat map of the metabolites. So this is according to the treatment condition, and then these are the metabolites that are increasing or decreasing according to their diet. And so through that, we're able to tease out what pathways are turned on or turned off. And then when we use a specific program to analyze that, we're able to see, for example, in intermediary metabolism, that the pathways in green are being turned on if you deprive the cell of serine, and the pathways in blue are being turned off. And that tells us that your cell metabolism really changes, and it turns on specific programs according to the nutrients. And we hope to take advantage of that to understand if we can manipulate metabolism to get rid of certain cancer cells. One final example I'm going to give you, which also required a lot of applied mathematics. This is one of my graduate students' work, and he is very interested in structure of proteins. And this is sort of a schematic structure of a protein. But what happens is, in order to get a protein structure, the protein has to be purified very highly and then crystallized by specific salts, and it becomes a crystal. And then you can diffract that crystal and obtain a lot of data on the way this crystal folds. And so this was actually done at Amgen, a pharmaceutical company. But then my student was able to, when he got this crystal structure, was able to use it to understand how this crystal will now be used to understand specific drugs that would inhibit its activity of the protein. So he's able, by mathematical docking, to show that a specific inhibitor will bind here that we know inhibits. And then he can model 1 million compounds, a library that has 1 million compounds. He feeds the structures into a computer with the structure of the crystal, and then he comes up with molecules that actually bind in the structure and potentially could be considered inhibitors or activators. So he came up with 85 molecules that docked in there and were, and then he tested them, and he found that there were several amongst them that are potential good, this is the control of inhibition. He found several that would be good inhibitors that we're able to use to modify the structure by chemistry, and then go back and develop an inhibitor for this enzyme that's very important for us in cancer. So I think I want to stop giving you examples, and I'm happy to tell anyone more details about them. I just wanted you to understand the incredible strength of what we're able to do using data, big data. And again, what I'm talking about today really is big data, but then we can distill it down to manageable data. It's nothing like what some of the things you're going to hear about, which are more like metadata. So big data is a hot and trendy term. It's here to stay, and it's only growing. Another term is metadata, which is data about data. And it's where now all the action is. And I think that's how you'll spend most of your day, and I'm really excited to hear and learn. The applications and implications to life sciences are immense, and the challenges are large, and many have emerged in data storage and in data governance, and I think those are going to be some of your topics today, which I will be keen to hear more about. And I'd like to thank you for your attention, and thank AUB and WiDS for this really nice invitation, and my university for support of a lot of this work. Particular people, I would acknowledge all my students, and thank you for your attention.
Thank you very much. We're going to defer the break, if you agree, until 12 o'clock. We'll have a larger break time. We just have time for 1 or 2 questions from the audience, and then we'll proceed with the second speaker.
We can have more discussion later.
Yeah, okay, one question.
Oh, go ahead. Ladies first.
Sometimes when we do research and we come up with data that's against the benefits of the pharmaceutical companies, in this case, what do you do? Because we know that sometimes we do research and then we figure out that, for example, the cure for cancer isn't like costly like the existing now medical treatments. But we face problems with the company that are actually buying those kinds of medical requirements. So what do you do in this case?
So that's an interesting problem. Of course, there's always going to be resistance from pharmaceutical companies because they invest a lot of money in bringing a drug to fruition. But I think, I mean, the ethics of the problem, it's, you know, one has to be ethical about their ability to understand and use and say things, speak up to things. I think this happens less, maybe I shouldn't say that. I hope it happens less and less. But also, one interesting caveat to that is that because of precision medicine now, we're finding that some drugs that are good for one patient are not good for the other patient. And so if we're able to take advantage of that and use that to our benefit and say, okay, maybe 1 out of 100 patients benefits from this drug, but that's a good drug for that patient. So that's the goal of this precision medicine approach. And so we have to always be vigilant.
Thank you.
There's one in the back.
Over there.
Yes, thank you for the presentation. Basically, my background is in philosophy, basically in philosophy of science. And I'm a little bit skeptic on the kind of justification of the data, of the methodology you're using. Basically, I'm not saying that you're not doing a great job to data mine the results, but my question directly is how to justify it and how to know that it's not just another paradigm or not just how to falsify it. I mean, we might just be generating new data which is telling us something which is not true about the test itself. How can you be true about the data you're generating?
I think I think there is a lot of data out there, and you have to be aware, and you have to always set out to prove yourself wrong, and then rather than try to prove yourself right, because then and I teach my students, I'm like, try to set out to prove your hypothesis wrong. But then when you obtain the data, I mean, if it's hard data, like you're sequencing a protein that you found from a patient with cancer, and you find that 7 out of 10 patients with that cancer have this protein really high, and then turns out this protein has a specific function that when you inhibit it, it kills the cancer, you've sort of validated that story. So you always you get the big data, but then you have to drill down and validate them. So I think, at least in the biomedical field, that's very critical. And that's how we do it. Okay.